Molecular Biology and Genomics final presentation. I'm Danielle Moore.
I'm Kayla Hicks.
And I'm Lauren Banks.
Okay, so what is HMPCC? HMPCC, or hereditary non-polyposis colon cancer, is a hereditary disease with a higher risk of developing colon cancer. A mutation in gene MSH2 is linked to HMPCC, and some common symptoms include fatigue and random weight loss. So the mismatch repair mechanism removes bases that are incorporated incorrectly during DNA replication, and US and UL recognize the DNA damage. It's considered exonuclease activity, and DNA polymerase produces the new strand. Ligase connects the newly synthesized DNA strand to the existing strand. So for our first module, the method was digestion of the plasmid to determine which E. coli strain contained the PMSH2 plasmid. Our independent variable was the size, or how many base pairs of the band was produced and visible on the gel, and the dependent variable was which plasmid each digested product contained, MSH2, MSH2 prime, or PRS41. So first in this procedure, we took 3 20 microliter digestion reactions, and those were made, and each had 500 nanograms of plasmid DNA, fast digest buffer, X-ball and A-ball. And then the 3 20 microliter control reactions contained fast digest buffer and 500 nanograms of plasmid DNA. The mixtures were then incubated for 30 minutes at 37 degrees Celsius, and both the undigested and digested products were stored in the freezer. So that would be 6 total products stored in the freezer.
After plasmid DNA isolation, SP0 had a concentration of 326.3 nanograms per microliter and a ratio of 1.85. SP1 had a concentration of 769.8 nanograms per microliter and a ratio of 1.89, and SP5 had a concentration of 363.6 nanograms per microliter and a ratio of 1.85, as shown in Table 1. After digestion of plasmid DNA, strain SP0 produced one band around the size of 5,000 base pairs. Strain SP1 and SP5 produced 2 bands around the size of 4,900 base pairs and 5,000 base pairs, shown in Figure 1. The predicted size of the 2 fragments of PMSH2 made after restriction digestion by X-ball and A-ball was 4898 base pairs and 4427 base pairs in Figure 2b. The predicted size of the 1 fragment of PRS413 that was made after restriction digestion by X-ball and A-ball was 4898 base pairs and 72 base pairs.
So to discuss the results that we just saw, prior to beginning the experiment, the predictions were made about what the outcome would be of the experiment to determine which strains contained which plasmids. And the plasmids that we were testing for were PMSH2 and PRS413. The plasmids were digested using the restriction enzymes X-ball and A-ball, and after digestion, the images that we just saw on the last slide showed that SP0 had produced a band around 5,000 base pairs, and SP1 and SP5 both produced 2 bands around 4,900 and 5,000 base pairs. And so after comparing the observed base pairs and our predicted base pairs, we concluded that SP1 strain and SP5 strain contained PMSH2 and SP0 strain contained PRS413 plasmid.
Okay, so for our second module, the method used was digestion of the PCR product, and this was done in order to determine which plasmid DNA contained the mutant MSH2 insert. So the independent variable again was the size of the bands or the base pairs that were visible on the gel, and the dependent variable was which digested product was mutant and which was wild type. So 320 microliter samples of the digestion reactions were made that contained 1x REHAE3 buffer, 2% REHAE3, and 10 microliters of PCR product. 3 more samples were made, 20 microliters, of the control reactions of undigested products were assembled containing 1x RE buffer, 10 microliters of PCR product. The reaction was incubated at 37 degrees Celsius for an hour, and then the 3 digested and 3 undigested products were stored in the freezer.
The name of the restriction enzyme used in the lab was HAE3. Lane 1 contained the 100 base pair DNA ladder. Lane 2 had sp0 contamination. The digested PCR products of sp1 and sp5 were loaded onto lane 3 to 4. The undigested PCR products of sp0 contained contamination. sp1, sp5 were loaded into lane 5 through 7. The observed band sizes for sp1 digested PCR products were 800 base pairs and 125 base pairs, which were similar to the observed sizes, which were 763 base pairs and 120 base pairs. The observed band size for sp5 was 900 base pairs in Figure 4. The observed band sizes for sp5 digested PCR products were similar to experimental sizes, 883 base pairs. The predicted size of the 2 fragments made after digestion of wild-type PCR product was 763 base pairs and 120 base pairs in Figure 3C. The predicted size of the fragment made after digestion of the mutant PCR product was 883 base pairs.
The results of the digestion of the PCR product were pretty straightforward, but just to reiterate what was used, there was a specific restriction enzyme, HAE3. It was used to digest our PCR product specifically because it can identify our mutant strain, which was SP5, from the wild type MSH2 strain. And prior to performing the experiment, we had made predictions about the wild type strain and the mutant strain. And so what we saw in the last slide was digested and undigested strains pictured next to the wild type MSH2. And from the results, we concluded that they were very similar. Our observed results were very similar to the predictions that we made. And from this, we concluded that the digested products of SP1 were wild type PMSH2, and the digested product of SP5 was the mutant PMSH2 prime.
So our third module used the method of yeast transformation. The yeast were transformed, and the reporter strain AGY75 already contained PSH44 with the mutant PMSH2 plasmid PMSH2 prime. So as far as the independent and dependent variables, the independent variable were the essential nutrients in the media, and the dependent variable was yeast growth on the plate. So basically, the yeast could only grow if they had all of the essential nutrients. The yeast cultures were resuspended by pipetting, specifically pipetting up and down. And so yeast strain AGY75 transformed with PMSH2 or SP1 and the overnight culture with OD600. Yeast strain AGY75 transformed with PMSH2 prime, SP5, was the overnight culture with OD600 as well, and then yeast strain AGY75 transformed with PRS413 or SP0 was the overnight culture with OD600. And the OD600 basically equals the concentration of 1 for each yeast strain.
In Figure 5, it shows the yeast transformation of mutant plasmid. There were 10 colonies on the negative plasmid plate. The yeast was not able to grow on the SD plate because it was unable to produce all 3 essential nutrients. There were approximately 700 colonies on the positive plus plasmid plate.
And to further explain what we just saw on our last slide, the slide pictured on the left was the positive media, and the slide pictured on the right was the negative media. The purpose of yeast transformation was to transform the yeast reporter strain AGY75, which already contained the PSH44 plasmid, with the mutant PMSH2 plasmid, or PMSH2 prime. The yeast was not, as Kayla said before, the yeast was not able to grow on the SD plate, the media plate, because it was missing the 3 essential nutrients that yeast needs to grow. Excuse me. The PSH44 plasmid contains TRP1 and uracil-3, but the yeast would still need history for proper growth. So that is why the yeast needed to be transformed with the second plasmid, because it could not grow properly with the first one. And so the observations that we saw on the last slide show that the yeast plated on the left plate had been transformed with PMSH2 plasmid. PMSH2 plasmid, because there were over 700 colonies. Once it was transformed, it possessed all 3 nutrients needed for growth, and the plate on the right showed very little to no growth because it was missing its nutrients.
Fourth and final module, the FOA assay used the FOA assay was used to measure mutation rate in 3 yeast strains carrying PMSH2, SP1, PMSH2 prime, SP5, and PRS413 or SP0. The independent variable was the mutation rate, and the dependent variable was yeast growth in the presence of FOA. The FOA plate had an SD medium containing FOA, URA, and all essential nutrients except for histidine, tryptophan, leucine, and 309. The SD minus HIS minus TRP and minus URA plate had an SD medium containing all the essential nutrients except for histidine, tryptophan, 309, leucine, and uracil. 9 tubes of yeast suspension, so 3 concentration and 3 genotype, were made and centrifuged to remove most of the supernatant. The yeast pellet was resuspended, and then 50 microliters of the yeast suspension was pipetted onto the FOA plate, and another 50 microliters of yeast suspension onto the SD plate. The plate was left alone until the liquid dried, and when the liquid did dry, the plate was inverted and incubated at 30 degrees Celsius for 24 hours.
All 3 strains on the SD plate were expected to grow, and Figure 6 shows the growth. Under OD.1, SP1 had the least colonies as expected, SP5 had the second most colonies, and SP0 had the most colonies out of all 3.
FOA experiments can be used as a model to determine if a specific mutation in MSH2 may increase the carrier's chance of developing hereditary non-polyposis colorectal cancer, or HNPCC. The yeast colony growth was observed under different concentrations, as stated before, 3 different concentrations with 3 different genotypes of yeast. OD1, concentration OD1 had the most growth for all three of the strains, and the concentration OD 0.01 had the least amount of growth for all three of the strains. And the results observed concluded that SP5 had growth under OD 0.1, so the MSH2 gene is a pseudo wild type allele. And to conclude our presentation, this research is important to the real world because it can help to further research in gene mutation, cancer research, hereditary disease, and numerous other subjects. While furthering research, the end results will also include discovering and creating more preventative methods of medicine in the health field. We know that this yeast data is useful for human research because we have discovered the human cognate of our mutation, which was SP5 E. coli strain, and it was originally, the yeast mutation is located at M707I, and the human cognate mutation is located at M688I. And yes, I think that concludes our presentation.